this video, we will discuss about the mechanism of hypoxia on the cell surface receptor of CAR NK or NK CAR. So what we will discuss, how the hypoxia will affect the receptors on the CAR NK surface, and in the other figures, that is figure on the right hand side, we will discuss um, how these different receptors on the CAR NK plays a role. First, we will start with the figures on the left hand side, where we can see that the tumor cells uh, are shown on the left hand side and the CAR NK cells shown on the right hand side. And we know that most of the solid tumors will form a tumor microenvironment. So this slide is basically focuses on interactions of the solid tumors with the CAR NK. So these solid tumors will have a tumor microenvironment, and that usually has these conditions that are mentioned in the red box, that is hypoxia. It is hypoxia. Hypoxia means it will has a less amount of oxygens available than what is usually required, and this pH is also low because of the uh, uh, products secreted by the tumor cells, and the adenosines are high and the amino acids are also low. These are the usual conditions in the tumor microenvironment. So what will happen because of this hypoxia? This hypoxia will cause a decrease in this phosphorylations of the ERK or STAT3, or ERK STAT3 signaling pathways will goes down, and this will reduce the cytotoxicity effect. And at the same time, this mica granulase B will also goes down. This will cause reductions in the granules that are granules or such as this granzyme that are released by the CAR NK cells, or that can act on the tumor cells. That will also goes down, and the receptors that usually present on the CAR NK cells for the detection of the tumors will also goes down because of the hypoxia conditions. One positive thing happens is that hypoxia will trigger HIF, that is hypoxia induced factor, that will cause the upregulations of the glycosylases. However, the overall effect of the reduction in the cytotoxicity and the increase in the glycosylases is that the overall cytotoxicity effect of the CAR NK cells goes down. The glycolysis is also inhibited by the increase in the adenosine, and thereby what will happen, the amino acid, thereby what will happen, this amount of energy that is required by these CAR NK cells will also goes down because of the suppressions of the glycolysis. The glycolysis also been reduced through this upregulations of the lipid, and this upregulations of the lipid usually occurs because of the amino acids that are present in the tumor microenvironment are being consumed by the tumors, and that will cause the release of the lipids, and these lipids when that will then interact with the CAR NK cells, and that will leads to a decrease in the mTOR or CMIS uh, pathways, that will further cause suppressions of this glycosylases, and this suppressions of this SLC3A2 or SLC7A5 genes. So this is some of the effect that CAR NK observe when it comes in contact with the hypoxia conditions, or when it come in close or vicinity to the hypoxia conditions. And now we will discuss how, if the CAR NK cells does not have to go through this hypoxia rate, or somehow it will overcome these tumor microenvironment conditions, how it will act. So this CAR NK cells has these receptors, that is NKGD2, that can interact with this NKGD2L ligand that are present on the tumor cells. It is similar to the major histocompatibility complex in the T cells, CAR T cells or the T cells. This is about the CAR NK cells. So once they interact, then it will cause a activations of this DAP10, and that will lead to the upregulations of the pathways such as P13K, P85, GRB2, VAV1, and that will leads to the increase in the cytotoxicity. At the same time, this DNAM1 can interact with these ligands that are usually expressed on the tumor, such as CD 155 CD12, and that will also contribute towards increase in the cytotoxicity. This HLA interact with this KIR, LNS receptors that will trigger ITEAM and DAP12 activations, ITAM also, and we know that ITAM will also cause the increase in the immune response against the tumor cells. And this NCRL then can interact with the NCR receptors or NCR ligand, and these NCR receptors may NCR receptors can is NCR receptors include this NKP46, NKP44, NKP30 receptors. So NCR ligand can interact with any of these receptors, and all this will contribute towards increasing the cytotoxicity of the CAR NK cells against the tumors. So this is and these NKGD2 activations can also be supplemented by the cytokines that are released by tumor cells, such as interleukin 15, 10, 12, TNF alpha, INF alpha. So, and these small interactions that are shown here, both of them will, all of them will trigger these DAP10 activations in the intercellular part of the CAR NK cells that will cause the increased cytotoxicity effect against the tumor cells. So I hope you understood to some extent, or a brief idea about how this hypoxia helps in the suppressions of the CAR NK cells and how these receptors interacts with the tumor cells if there is no tumor microenvironment conditions are present. So, and these things are usually can be replicated in the in vitro conditions or in the lab scale conditions or in the ex vivo conditions. I hope you understood to some extent how this mechanism works. If you need to understand more about these mechanisms in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.